Hello, my name is Eddie Park. I am from the laboratory of Yixing at the Children's Hospital of Philadelphia. The title of my talk is Genetic Variation and microRNA Targeting of A-to-I RNA Editing, Fine-Tune Human Tissue Transcriptomes. Here is a brief overview of A-to-I RNA editing. A-to-I RNA editing is a biological process in which adenosines in RNA transcripts are deaminated to inosines. Inosines are structurally similar to guanosine and are read by many cellular processes as guanosine. Additionally, inosines in RNA transcripts are reverse transcribed to guanosine in cDNA. There have been many documented downstream consequences of A-to-I RNA editing, such as protein recoding, altered splicing, and changes to RNA structure and stability. This process is driven by the ADAR family of enzymes. In humans, most of these events occur in allorepeats, which are able to form long RNA hairpins and act as substrates for ADAR enzymes. Here is how we calculate RNA editing rates from RNA-seq data. First, we align RNA-seq reads to the genome. The inosines in RNA molecules are reverse transcribed to guanosine in the RNA-seq data. We calculate the A to G substitution rate from the aligned reads as a measure for the RNA editing rate. Here is an example of a Qtl analysis. This is an expression Qtl in which the gene expression of a gene is correlated to genotype. As shown in the illustration on the left, individuals with the G allele for the SNP express lower levels of the gene, while individuals with the A allele express higher levels of the gene. The middle plot shows a linear correlation between the gene expression level and the genotype. This type of statistical test can be applied to all genetic variants within a large genomic window as shown in the plot on the right. Then, the genomic polymorphism with the most significant association to the gene expression can be identified. In addition to expression Qtl, other types of quantifiable molecular traits can be studied with Qtl analysis. For this project, we looked at RNA editing Qtls, which are abbreviated as EDQTLs. This involves finding associations between RNA editing levels and genotype. We identified approximately 3,000 unique EDQTL events across 49 tissues. The plot on the left shows the number of EDQTL events in each tissue. The number in parentheses represents the number of samples per tissue. The plot on the right is an example of a significant EDQTL event in the FAM129A gene. We noticed that some tissues consistently have weaker EDQTL effect sizes for many EDQTL events. The two plots on the left show the EDQTL effect size can be variable for different tissues. The left plot, which has a strong EDQTL effect size, is from the nucleus accumbens, which is a region in the brain. The right plot, which has a weak EDQTL effect size, is from the skeletal muscle. To investigate this further, we plotted the effect size for this EDQTL across all 49 tissues. We found that the tissues expressing low levels of ADAR have weaker effect size. Additionally, we see this trend in many other EDQTL loci. We also see a similar trend in which tissue-specific expression of ADAR-B1 drives a subset of tissue-specific EDQTL events. In this example, an EDQTL in RAB-GEF1 is present in tissues expressing high levels of ADAR-B1. Here is an example of an RNA editing site that has been previously described in the literature that we also identified as an EDQTL RNA editing site. In this study, the authors show that microRNA 125A targets the unedited form of the DHFR mRNA. They were able to show that the RNA editing at this site is able to block microRNA-mediated degradation, and this results in increased steady-state levels of the DHFR mRNA. In our analysis, 3 tissues had an EDQTL for this site: LCL cell lines, cultured fibroblasts, and the spinal cord. The EDQTL plots for these are shown at the top 3 plots. In the EDQTL plots, we notice that there was a nonlinearity to the data. The red dashed lines represents a straight line in the data, while the blue dashed lines represents a quadratic fit of the data. We quantified this nonlinear shift and found that it correlated with the microRNA levels of miR125A. We also observed that there was an expression QTL at this loci. To help explain this mechanism, we created a simple simulation. For the data on the left, we simulated an EDQTL event in which the RNA editing level decreases across genotype. In this case, both the edited and unedited forms have the same stability. In this case, there is an EDQTL present, but the mRNA transcript levels are identical across the genotypes, so there is no expression QTL present. For the simulation on the right, the same EDQTL is used, but we also simulated a microRNA degrading the unedited transcript by 80%. So for this case on the right, the EDQTL signal becomes nonlinear and an expression QTL forms. So to recap, we observe a good concordance between the experimental data on the left and the simulated data on the right. We demonstrate that microRNAs can interact with EDQTL events to generate expression QTL by targeting the mRNA in an editing-specific manner. Since individuals with different genotypes would have variable RNA editing levels, this would result in variable steady-state gene expression levels for these individuals. Co-localization analysis is a way to compare 2 QTL datasets. In this study, the authors developed a Bayesian test to compare the summary statistics for 2 QTL signals to see if the 2 traits share a common set of regulatory variants. If the 2 traits are associated with the same regulatory variants, the posterior probability for hypothesis 4 would be close to 1. Here are Manhattan plots for the EDQTL and EQTL data for the loci and the DHFR gene that was previously discussed. The EDQTL Manhattan plots are on the top, and the expression QTL Manhattan plots are in the middle. The bottom plot represents scatter plots comparing the negative log 10 p-value for the EDQTL and expression QTL data. The value in parentheses represents the co-localization posterior probability between the RNA editing and gene expression. The high posterior probability suggests that the RNA editing and gene expression levels are regulated by a shared set of genetic polymorphisms. Here is an example of a loci that overlaps with an EDQTL, EQTL, and a GWAS trait. The EDQTL and EQTL plots are shown on the top left. RNA editing in the FADS1 gene is also linked to the steady-state mRNA levels of that gene. Additionally, the RNA editing and gene expression levels are also linked to the GWAS trait of total cholesterol levels. This suggests that genetically regulated RNA editing can alter the gene expression levels of FADS1 to influence total cholesterol levels in individuals. Next, we use a stringent computational pipeline to predict additional EDQTL, eQTL co-localization events that are mediated by microRNAs. And we were able to experimentally validate a subset of our predictions. Here we have our schematic model linking EDQTLs to eQTLs and complex traits. On the left, we have EDQTLs that arise from genomic variants that influence RNA editing. If a microRNA interacts with this EDQTL in an editing-specific manner, differential mRNA transcript levels can arise and result in an expression QTL. The resulting expression QTL could result in variations in complex traits. In conclusion, in this study, we identified 3,117 EDQTL events across 49 human tissues. We found that tissue-specific expression of ADAR and ADAR-B1 are responsible for a subset of tissue-specific EDQTL events. Lastly, we identified a novel mechanism in which microRNAs can interact with EDQTL events to create eQTLs and influence complex traits. Thank you all for your attention, and here is everyone that participated in this study.